Hello, everyone. Welcome to today's webinar. I'm Catherine Dold, and I'll be moderating today's event. Today's webinar is titled Adapting Well-Known Chromatographic Techniques for the Analysis of Biomolecules and is being sponsored by MacMod Analytical. CNEN works with sponsors to identify topics of interest and value to CNEN's audience and consistent with the mission of providing news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need any technical help, please look at the Help widget at the bottom of the screen or type your query into the Q&A box. If you get disconnected during the webcast, please log in again according to the instructions you received earlier. You're encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. Questions will be answered at the end of the presentation, and as your moderator, I'll be posing as many of them as time permits. Please note that CNEN does not endorse any company, products, or services that may be mentioned in the webinars. Also, each webinar will be archived at CNEN online after the live webcast. Our presentation today is being sponsored by MacMod Analytical. MacMod's mission statement is Smarter Chromatography. What does that mean? It means that when MacMod makes a recommendation to you about an HPLC or UHPLC product, it is to partner with you to offer innovative solutions to your complex problems. They use advanced state-of-the-art analytical software to match their product portfolio with your separation needs. They leverage their 30-plus years of experience in the LC column business, their technical expertise, and their manufacturing network to help you solve your toughest application problems. Our presenter today is Gemma Lowe, Senior Project Manager. Ms. Lowe is a member of the Business Development Team at Avantor UK and is responsible for leading a wide range of HPLC and UHPLC training events and seminars across Europe. She has more than a decade of experience working in the HiChrom technical support team, troubleshooting chromatography methods, recommending suitable column chemistries, and supporting the vast range of HPLC and UHPLC columns in the portfolio. Originally a biochemist, her focus areas include the analysis of biomolecules by various chromatographic techniques and the analysis of polar analytes. I'll now hand the program over to our speaker.
Thank you very much, Catherine, and a very big thank you again to MacMod for inviting me to speak and to talk about something I'm very passionate about, which is the analysis of biomolecules. So thank you, MacMod, for your partnership for all these years. We have quite a busy agenda, quite a lot of topics that I hopefully can cover with you today and leave some questions at the end, or some time for questions at the end. We're going to start with a bit of background, what are we talking about, what are biomolecules, and then really look at techniques that we're really used to for small molecules and how we can adapt them to the analysis of larger molecules. What things do we need to think about? What do we need to consider really? How is biomolecular analysis different to that analysis of small molecule drugs, for example? So all of these sections, we're going to look at different parts of your method that need to be modified perhaps, or at least considered when you're analysing large molecules, proteins, peptides. That's going to be the main focus of our agenda today. So as I said, lots to cover, so we will go through these slides quite quickly. So biomolecules, and not everything we're going to talk about is strictly a biomolecule, because we have got some synthetic molecules. Normally we call a biomolecule something that's been made in a living system, but actually we can synthetically make peptides. We synthetically make oligonucleotides, but they are sort of loosely classed as biomolecules. So we're going to talk about how we can analyse those chromatographically. And one thing all of these molecules have in common is that they're very complex. So proteins, peptides, monoclonal antibodies, antibody drug conjugates, plasmids, vaccines, they are large, biological molecules that are very complicated. They have a primary amino acid structure in most cases, and that then folds up to give more structure, secondary, tertiary structure. And when we want to use them, whether it's for biotherapeutic use or, if you're working with biological washing powders, they've got proteins in there. So we need to make the protein that we intend to make. The problem is when we're generating them in a living system, they turn out to be very heterogeneous. So, we need to properly characterize these very complicated molecules, and chromatography is a wonderful tool to be able to do that. Some of these different forms can potentially be harmful if we have, for example, a lot of aggregates in our mixture. Then that could cause a reaction if you're using your protein as a biotherapeutic. So we need to make sure we don't have a lot of aggregation. And some of the modifications that we want to characterize could increase the aggregation. So that's just one example of many things that can happen within our system. So we want to characterize all these heterogeneous versions of our protein. We need to look at degradation products like we would for a small molecule analysis, processing variants. And when you've got these really complex mixtures, you don't just have a handful of processing variants, you can have hundreds or thousands of different variants, especially if you're working with very large molecules. So very small changes in that basic amino acid structure of a protein can affect the safety and the efficacy of a drug. So it's imperative that we monitor any changes. And chromatography is a fantastic tool to do that. 95% of the protein is going to be the same in all of these variants. We just have to try and find that 5% that's differing. So we're going to have a look at how chromatography can help us with that. Now, biochromatography is the same as small molecule chromatography. We have something we need to purify. We have our analytical method. And what you'll see is there are many different modes we could actually utilize for biomolecules. And then we have our external factors. So it's not significantly different from analyzing aspirin. It's not, it's just potentially more complex because you have such a large protein that has all these different side chains, meaning it can have lots of different functionality. So HPLC is very well suited to biopharmaceuticals. It's a really well-established, well-understood technique, and it will work for the vast majority of proteins and peptides. So all of the great development that we've done on small molecule HPLC, we can translate to biological molecules. So as I mentioned, these biomolecules, they're produced in a living system in most cases. They haven't historically been characterized as well, but more recently, there's a lot more legislation, there's a lot more guidance that says we need to really characterize all of these different variants of this protein, because any errors can cause problems. I mean, we're using these biotherapeutics as the majority of our drugs. So I think 6 or 7 of the highest selling pharmaceuticals are biomolecules. Obviously, there's insulin in there. That's always been well established. But now we have many more monoclonal antibodies or monoclonal antibody-related biotherapeutics. So it's a very big industry. More recently, some of these are coming off patent. So we've got biobetters, biosimilars, imitator drugs that everyone's trying to manufacture. And we need to check that we are actually characterizing all of these to the best of our ability. So in order to use these pharmaceutical APIs, the purified compound must be available, and a method for analysing it and accurate quantification is necessary. So there's ICH development guidelines, Q2A, Q2B guidelines that look at the safety and the efficacy, and then we can demonstrate that to the regulatory bodies. So we need to make sure that we're proving product purity, we're monitoring production to make these biotherapeutics useful and safe for everyone. So when we think about a protein or a peptide, it comes down really to that initial amino acid structure. These are all made up of amino acids. And, the difference between a peptide and a polypeptide and a protein is just the size. So peptides tend to be short chains of connected amino acids, really with not a lot of secondary structure. Then when you start to get polypeptides, 20 to 50 amino acids in length together, you start getting some secondary structure forming. And then 50 plus amino acids, we class that as a protein. And these have more structure. They potentially are folding into alpha helices, beta sheets. And that is great for their biological activity. But as a chromatographer, it makes it more challenging to see if there's a change in one amino acid that could potentially be folded up into this very complex high molecular weight structure. So when we're looking at different chromatographic methods, a lot of the time, if we want to see all of the changes in our protein, we need to unfold it. We need to expose those side chains that are folded and hidden within that tertiary structure. You can do this with an intact protein, or you could digest it and look at the individual peptides in a peptide mapping experiment. And both of those we can utilize chromatographic methods for. Now, I really don't have time to go through all of the potential modifications that a protein can undergo. But there's a list of some mentioned here. All of these are relatively common modifications that can happen naturally or during the actual manufacturing process of a protein. For example, with a monoclonal antibody, we're commonly expressing those in mammalian cell culture. Then we purify the antibody with lots of chromatographic steps and filtration. We'll look at affinity chromatography, ion exchange, probably some hydrophobic interaction. And that resulting high purity bulk drug substance will still contain different variants of our antibody. And that's based on charge, size. They can have one amino acid difference. So all of these post-translational modifications, we call them, can occur by degradation during that the process of growing them. So we have to make sure that what we've made at the end is actually what we intended to make. Because you can very easily change one amino acid in that sequence and cause a very significant change in its biological activity or its efficacy. And therefore we need to make sure we're characterizing all of these modifications. So some of these modifications will change the charge on the protein. For example, the amidation. Here we've gone from an asparagine amino acid to an aspartic acid. So now I've got an ionizable side chain on one amino acid in my 100 amino acid length protein. But chromatography is so powerful, we can actually use it to separate that from the original protein that we were trying to make. And that's really why it's finding its feet for analysis, especially in the kind of QC or reaction monitoring of biological pharmaceuticals. So lots of different variations. Sometimes we need to denature the protein, unfold it to be able to see these changes. And if you're thinking about your chromatographic method development, think about how you want to analyze your protein. Do I want to look at my intact protein in its biological native form, or do I want to find out more, in which case I've probably got to denature it? So really, there's a couple of the most common techniques really we use. We can either have our completely intact protein and try and chromatographically analyze that. And actually, we can use reverse phase for this. Most of the proteins are quite hydrophobic, so you will get retention in reverse phase. But if you want to see more within that folded structure, then you probably have to denature the protein to unfold it, and then you can actually get more information about that protein structure. The other technique, and I've simplified this very much by just writing denature and digest, is to do some peptide mapping. So we actually chop up the protein into much smaller peptides and then run these chromatographically. And this really is most commonly run by reverse phase and normally connected to a mass spec. So these peptides behave a bit more like small molecules, and therefore we can run the nice peptide maps, get lots of peaks in a separation window, and get a fingerprint of that specific protein. So these ICH guidelines, as I mentioned before, peptide mapping is one of those in Q6b. So it's a critical quality attribute, and we need to confirm that primary amino acid sequence of our protein, if we're using it for therapeutic use, of course. So this will have to be done, especially if you're looking to launch a new biosimilar or biobetter. You'll have to make sure you run peptide maps for your final product. And you can then also compare those with the originated drug, the originator protein, to see where it differs in the amino acid sequence. So it's a nice way of comparison to the original protein. And there's lots of databases now available that show peptides as well as some of the other things we're going to look at later on, like the modifications. So I've mentioned reverse phase, and really that's what I'm going to talk about most during this session. But proteins really have a lot of chromatographic modes that you can use. Because they've got all these amino acids with different functionalities, there's the potential to use ion exchange, for example, because you've got ionizable side chains in there. Your protein will have a pi, and that's when you have equal positive and negative charge on your protein. But below the pi, you have a net positive charge, and above the pi, you have a net negative charge. So you could use either mode of ion exchange to separate out proteins if you've got different ionic variants in there, different charge variants. You can use size exclusion for aggregates, because size exclusion is a really good technique if you've got a significant difference in molecular weight. And obviously, if your protein is sticking to other proteins, you've got double the molecular weight for a dimer, you've got triple the molecular weight for a trimer, so you can nicely use size exclusion for aggregates. What I'm going to talk about mostly today is reverse phase, because we're adapting those modes that we know well for small molecules to our large molecules. And really, we don't use affinity or hydrophobic interaction with small molecules at all. They tend to be used mostly with proteins. So we're going to talk a lot about reverse phase and a bit about Hillock for more polar components and some of those post-translational modifications, specifically glycosylation. So reverse phase, as I've mentioned, is something we use a lot with small molecules. Well established, we understand it, we understand the mechanism, and you can use the same types of columns for proteins and peptides. The mechanism with proteins is a bit different though. It's not the same speed of separation that you have with small molecules, and it does act a bit differently. In theory, the most hydrophobic protein will elute last, the most hydrophilic protein will elute first. It's good if you've got small to medium molecular weight proteins. So as I'm saying here, up to about 20 kilodaltons. If you've got really big proteins, if you've got things like viruses, then reverse phase probably isn't the best technique for those. But for our peptides, for our small proteins, it works quite nicely. It's different from small molecule reverse phase because it's not the same partitioning that happens. So you've got a small molecule and you've got a C18 column, you tend to get a lot of partitioning and interaction of the small molecule with those hydrophobic C18 chains within the column. And that happens all the way down the column. When you have a protein, it's much more an adsorb-desorb, on-off type mechanism. So in theory, we should get really nice, sharp peaks with our methods because you just get a touch on the surface and then a desorption from the surface. But when you've got a very complex molecule like a protein, we don't have the very simple hydrophobic interaction. You have the potential for a lot more ionic interaction, hydrogen bonding, other mechanisms in there that actually interfere and don't give us that nice sharp peak shape. Hopefully if I scroll on to the next slide, you will understand this a bit further. So what I've got here on my graph, on the x-axis, I'm showing the percentage acetonitrile. And on the y-axis, I've got the log K. So that's the log of the retention time. So as you'd expect in a reverse phase method, as you increase the acetonitrile, you decrease the retention. With a small molecule, like benzene, that's our example small molecule here, you get this classic slope, gradually decreasing retention as you increase the acetonitrile concentration. When you look at a protein, so one of these much larger molecular weight compounds like cytochrome C, you get a much steeper slope. It's very much a small change in acetonitrile leads to a very dramatic change in the retention time. So these ballistic gradients that we love for our small molecule UHPLC applications do not work particularly well when we're working with proteins, because any small change in acetonitrile could lead to a very big change in that retention. So it's much better to work with shallow gradients for our protein separations, and you can narrow the range. So instead of running 5 to 95, you can run a much narrower range to actually get some time back, because otherwise you're going to be running your methods for a very long time at a very shallow gradient. So hopefully this gives you an understanding really of why we run at very small percentage increases of our acetonitrile when we're looking at large molecules. The main reason for this is that proteins are very slow at diffusing, and therefore, and that mechanism, that on-off mechanism, because when the percentage AUC, or the percentage of acetonitrile, gets to a certain point, instead of having that protein adsorbed on the column, it's desorbed. And you can see that in my graphics here. So here is my drawing of a protein, and you can see at the bottom here in the red, these red amino acids are the more hydrophobic amino acids. So they've clustered together in the protein to make a hydrophobic foot. And those will adsorb onto my chromatographic reverse phase C4 column. So they're interacting with the hydrophobic surface until I get the level of acetonitrile that makes them desorb from the column. So it's an on-off mechanism that happens at the top of the column. If there's no room for my protein at the top of the column, it will go down through the column till it finds a spare site. But in general, you don't need really long columns because your protein will just stick at the top and then come off and elute to give you a peak. So in theory, that should give us a nice sharp peak. But if you look at the structure of your protein, you look at all the other potential interactions that you could have, then you do get more distorted or broader peaks because there's other parts of the protein that can interact with the column as it goes down. Obviously, we want to minimize those. There's different things you can do to try and minimize any other additional interactions in the column. But really, we want to just have that hydrophobic on-off interaction. Peptides, they have a more similar mechanism of interaction with the column to a small molecule, but they do tend to be a bit like a large molecule, a bit like a small molecule, depending on their structure and folding as well. So it does make sense to have a longer column for peptides, but for proteins, unless you want a higher loading for purification, for example, then you can get away with quite a short column length, save some time. So as I mentioned, one of the other techniques you can use for protein analysis is to digest your protein and analyze the MAP peptides. And I have simplified it very much by saying denature, reduce, alkylate, digest. That's your rate determining step. The chromatography really won't take as long as the actual preparation of your peptides. So you could potentially have a longer chromatographic step because it can take you a day and a half to prep the sample before you even get it onto the LCMS. But there are better techniques now than when I was at uni and had to spend lots of time prepping these proteins. So there are kits you can get available now to help speed this up. Most of these are based on trypsin digestion. So trypsin cleaves the carboxy terminus of lysine and arginine residues. So it will cut at a certain known amino acid, and therefore you can use these maps to look at the actual structure, that primary amino acid sequence of your protein, and compare it with what you were trying to make, or with maybe a reference protein if you were looking to make it different from that. We can also help to identify where some of the secondary and tertiary structure might be. You could look at possible sites for glycosylation. You can look at different post-translational modifications using the peptide map. And as I've mentioned before, you tend to couple this to mass spec. High resolution mass spec is best because then we can get more information. These are very complex co-eluting peaks most of the time. So mass spec gives us that extra level to help separate them out. When you've got such a complex mixture, reverse phase isn't always enough. So the 2D analysis using maybe ion exchange, or more commonly now Hillic, is favoured because then you can actually get a bigger separation of some of those more hydrophilic or potentially charged peptides. So you can couple these together to get more information. And here's just an example of what these look like, these peptide maps. It is part of those ICH guidelines, and it is part of the multi-attribute method as well. So for anyone who's making biological pharmaceuticals, then it will be something you have to do. So C18, again, is probably the most common, but coupling that with something with a bit more selectivity can be of benefit. And that leads me nicely into selectivity, which is the first sort of area I wanted to talk about when you're thinking about developing a method for your proteins. And it actually comes back to the same factors we think about for small molecules. So what type of column am I going to use? The addition here is that we need to think about porosity. We need to think about how big the holes in our silica or our polymer are when we've got these really big proteins, because most of the surface area in a column is actually within the pores. So if your pores aren't big enough, your protein can't enter them, and you're actually negating all that surface area. Other things like pH, percentage of acetonitrile, type of buffer, column temperature, those all need to be considered. And most of the time we're going to be running a gradient, so all of the factors on the screen are really of interest when we're running our protein methods. When we're talking about our gradient, as I've mentioned, a nice shallow gradient is good, but also optimizing the flow rate can be of benefit. Now, large molecules are very slow to diffuse, and if the flow rate is too fast, you can have issues, meaning that you don't get the best method. There's not enough time for that protein to interact with the column, and therefore optimizing the flow rate can be much use in your method development. So do consider the flow rate more so in large molecular weight analyses than you would for small molecules. If you go too fast as well, you can cause frictional heating and aggregation. So just do consider that with the flow rate as well. So here's my example of why you need to consider the pore size of your column. I've got 3 analytes here, nicely coloured in green, blue and red. The small molecules can easily enter the pores, those green ones. The blue molecules can partially enter the pores, but it does depend on the orientation. So actually it's not just the molecular weight that you need to think about, it's the hydrodynamic radius. So how they're folded, what shape and size they are when they're in the form that you are going to analyse them in. I was going to say native form, but actually when we're running a reverse phase method, often we've got a lot of acetonitrile in there, and that will inherently denature the protein. So it might not be in its fully folded native form. So we need to consider that hydrodynamic radius of our protein. This red protein here, no way that's going to fit in the pores. Therefore, it's lost the potential surface area inside those pores and will come out quite early. So you need to consider the pore size of the column dependent on the molecular weight and hydrodynamic radius of your protein. And there's a bit of a rule of thumb here. If you're working with anything below 5 kilodaltons, the 100 angstrom porosity should be okay. If you've got something larger than 5,000 daltons, then 300 angstrom is kind of the classic size pore, and that will work for those larger proteins. It does depend on the pore geometry as well. So not every manufacturer will have the same size pore, and they won't all have the same shape pore. So even if it does work on one 300 angstrom column, it might not work on another 300 angstrom column. So do consider looking at different columns when you're doing your method development. Just another thing to mention here, with our protein analysis, we tend to use a C4-300. It's slightly hydrophobic. It will interact with that hydrophobic foot on the protein. When you go to a longer alkyl chain, like a C18, for example, you might get too much interaction with the column, and that actually kind of causes on-column denaturation of your protein. So your protein's starting to unfold, more of those hydrophobic residues are interacting with the column, and then it makes it very challenging to get a good peak shape because you've got different types of protein in the same column. So C4-300 for proteins. C18 is really good still for our peptides, and you can use those small 4 sort of 100 angstrom pore size columns for those. If you are working between, say, 20 and 50 kilodaltons, the 300 is going to be really okay. If you're going between 50 and 100 kilodaltons, it will work for some proteins, but it won't work for others. So on my previous slide, I showed the hydrodynamic radius of IgG, for example, so a typical antibody. That's 75 angstrom. That sounds like it will fit in a 300 angstrom column, but actually, in most cases, you will need a 500 or 1,000 angstrom pore size if you've got intact antibodies, for example. So you might fit 50 to 100 kilodaltons down a 300, but if you're going much bigger than that, I'd look at a 500 or a 1,000 angstrom porosity. So here's just a very simple application, some medium molecular weight proteins. I think the largest one here is 66.5 kilodaltons, which is BSA. And you can see with the different column chemistry, you get a slightly different selectivity, slightly different retention times. These are all straight alkyl chains. So actually, if we'd run these on a phenol, for example, that phenol would actually have probably given us a bit of a different selectivity with that added aromaticity. So that's a nice alternative to the C4 to have a look at as well. With peptides, we actually run peptides down our standard small molecule screening platform, which includes these novel stationary phases we developed with these additional mechanisms of interaction. So if you've got aromatic side chains, they can interact with pi-pi. If you've got more polar hydrophilic peptides, then actually it's a good idea to work with something that's got a polar embedded group, for example, like the C18 amide. And when you look at the peptide screening run, again, very simple peptides, you can see peaks starting to move around a lot more. So there's a lot more potential for different selectivity because peptides are a lot more similar to small molecules than our big intact complex protein. But it's definitely worth evaluating different stationary phases, even though most people will end up on a C18. And if you look through the literature, most of the peptide applications end up on the C18 column. We've done a lot of work with neuropeptides on some of the aromatic astationary phases. So they work really nicely on those early eluting peptides. So if you've got problems with peptides eluting early, then try something that's got a bit more polarity than a C18, because that will help to gain selectivity and gain retention for those polar peptides. If it's still too polar, you can try something like ion pair chromatography. And actually, all of the chromatography we do with proteins has some sort of ion pairing. PFA, which is our most common additive, is mildly ion pairing. So actually, most of the methods we use for these proteins are ion pair, which is a variant of reverse fate. It's great because it gives us more retention for our more polar analytes, but it also really improves the peak shape for proteins and helps with solubility as well. Some of the issues we have are that these aren't all mass spec friendly, but a lot more development has gone and interest has gone into looking at ion pairs that are mass spec friendly. One of the areas we've seen this is really advantageous is when we're looking at oligos. So when we're looking at oligonucleotides, you know, you look at that structure and you see some very polar groups on an oligonucleotide. When you've got a highly charged, highly polar compound, reverse phase isn't going to work, because reverse phase is based on retaining things based on their hydrophobicity. And I've got a very hydrophilic compound here that I want to analyse. So the only real way to get good retention and good separation of oligonucleotides is to add an ion pair reagent in there. So when we're looking at oligos, we're talking about short lengths of DNA and RNA. And there is a lot of interest in these for therapeutic uses, for genetic disorders, for cancers, antisense oligos. You know, it's a really hot topic at the moment. So to get good retention and good separation, because we make these and we want a certain length of oligonucleotide, but a lot of the failure sequences will be only one nucleotide different, one base different in length, and therefore we need a good way to separate those out chromatographically. So ion pair has proved really good at doing this. There's lots of different ion pair reagents that you can use. You need a hydrophobic, positively charged ion pair reagent, because then that allows the highly negatively charged oligo to interact with that hydrophobic stationary phase. So we can then easily separate the target oligonucleotides from their chemically modified byproducts based on hydrophobicity. So we can use ion pair, this variant of reverse phase, to get N+1, N-1 separations. You don't need mass spec, then TEAA is really the most common additive, the most common ion pair we use. And that is really historically what everyone has used. More recently, we've been trying to couple these to mass spec. So HFIP is a more mass spec friendly, if not person friendly solvent additive that we're going to put in the mobile phase. That improves electric spray desorption, fast evaporation, better ionization of the oligos. So it really is beneficial to use that in as low a concentration as possible. We've done some studies with TEA or hexalamine, or as you see here, dibutylamine. So there's lots of variants depending on what type of oligo you're looking at, and whether also the chain lengths as well. So some of them work better for different chain lengths of oligonucleotides. You can then scale this up. This is going back to the TEA application, but you can scale these up if you want to purify your oligo in the future as well. So with a prep column, you can actually collect the fractions quite nicely down to N, as I say, N+1, N-1 levels. Another alternative for more polar compounds, and really where it's found a lot of use, is for post-translational modifications like glycosylation, is HILIC. So HILIC is another mode of chromatography, separate from reverse phase, but of great benefit if you've got really polar compounds or ionizable compounds. And it is more mass spec friendly than ion pair. It does have a long equilibration time, but so does ion pair chromatography. There has been a lot of work trying to get HILIC to work for oligos as well, but that's not quite there yet, I think. So HILIC is not a new technique. It's been around since 1990 when Andrew Alpert coined the phrase. And it works by retaining more polar analytes. Really been used a lot for small molecules, but I have seen some work for glycopeptides, glycoproteins as well. So you could consider it if you've got the right pore size for larger glycosylated proteins intact. Well, we've seen that it's really of use is when you actually look at cleaved glycans. So most proteins, in fact, I think about 60% or 65% of the protein therapeutics on the market are glycosylated. That means that we need to look at that glycosylation pattern, that sugar pattern, and map it and make sure we're characterizing it properly. Again, it's one of the ICH D6B regulations, so you need to look at this as a critical quality attribute. I've got a very simplified diagram of how you can do this. Basically, cleave the glycans from your protein. In this case, it's a monoclonal, and we're going to use PNGase, which is a common enzyme we're going to use to remove those. We're then going to we're going to isolate them, then tag them with a fluorescent label, and then run through the mass spec with a HILIC type application. So it's very simplified, and again, this sample prep part really is the bit that takes the time. So that's going to be your rate determining step. When you've done that, you can get a nice pattern of what those glycans look like. You can check whether they're the right ones, whether that branched glycan is what you plan to have on your monoclonal antibody. And they're really important because they actually signal to the cell about how that antibody can get into the cell. They're important for cell clearance. They can cause a problem if there's the wrong type of sugars on there as well. So we really need to characterize that glycosylation pattern really to the best of our ability, because if you've got high mannose concentrations, it can cause clearance from the blood, and therefore obviously your half-life is not what it should be. If you've got glucose in there, that can contribute to ADCC activity. So it really is important to look at what type of glycans are on your protein. And HILIC is a great way to do that because it works on this on the opposite basis, the reverse phase, that the more hydrophilic compounds are retained and those more hydrophobic elute early. So it gives you the opposite elution order to our reverse phase. This is why it goes quite nicely with reverse phase as well for peptide mapping, because you can see more of the polarity scale. So I mentioned pore size. We need to look at the pore size of our columns when we choose our column for our application. You can see here, for larger molecular weight compounds, you get very broad peaks. So the y-axis is the peak width. You get very broad peaks if you use too small a pore size. And that's just because you haven't got access to the pores for those large molecules, and therefore you're not giving them the time for that on-off mechanism. You've got broad peaks and not good chromatography. So it's worth looking at the different porosities. When you're working with proteins, they tend to go down a 300 angstrom column, as I mentioned. You've got a very shallow gradient here. You can see just from 18 to 28 minutes, you're literally going 4% B increase. A very shallow gradient for our proteins. Medium molecular weight proteins, again, you can start to see some separation of the hydrophobic variants here even. Maybe they're different glycosylations because that changes the hydrophobicity. Maybe the sample started to degrade a bit. Reverse phase is really powerful at pulling out these things, something you're not going to get from size exclusion, for example. And this is just looking at a study one of our partners did, looking at 2 different pore sizes and 2 different chemistries. So here they were working with a small molecular weight protein, peptide in this case, only 2,500 Daltons. And they were looking for purification. So they ran all 4 analytical columns first to see which pore size was best and which chemistry. And in the end, they chose the phenol chemistry with 100 angstrom, which is actually what we predict for a that size molecular weight peptide, and got very good purity of their final peptide. We also need to think about the mobile phase, and really, acetonitrile is the most common mobile phase used. It works nicely. Methanol, there isn't much point in trying it for proteins and peptides. Acetonitrile really is the front runner when we're running these types of proteins. Could try ethanol if you're going for large scale purification, because it has got that viral inactivation. And also, if you're starting to get really strong hydrophobic protein interaction with your column, so really broad peaks, you could consider putting in 1 to 5% IPA, because that might break some of those hydrophobic interactions and help improve the peak shape. In terms of modifier, we use TFA most of the time. If you're using a high purity column, you can use less TFA. If you're using an older generation type low purity column, you can't even get away with 0.1% TFA. So the higher the purity column you're using, the less TFA you can get away with, and then that's obviously better for mass spec. And again, you can see here just some other examples with different concentrations of TFA. If you want to run mass spec, then we've tried things like formic acid as an alternative. Doesn't give you as sharp a peak shape, and it does change the selectivity. So yeah, there is an option to try formic acid as an alternative, but we've really seen TFA to be the most beneficial for peak shape. If you can use a higher purity column, obviously you can give less. You can have a lower concentration in your mobile phase, and that's obviously a benefit. In terms of other things we've tried, some of the perfluorinated additives can give you quite a different selectivity and also different sensitivity. So for peptides, for example, it's worth maybe considering some of these alternative additives. So things like acetic acid, formic acid and HFBA. Sounds a bit more complicated, but if you have a really challenging separation, those are the sorts of additives that you could try. You also need to look at the flow rate. So time's of the essence, obviously, and I've just seen the time on this webinar. So obviously we want to get things done as fast as possible, but when you're a protein, fast isn't always the best. So in this example here, we had to actually make this, we couldn't model it. We had to model it because it's very hard to get a true hydrophobic separation. So these small molecules here, for example, xylene, you can see a nice classic Van Diemter curve that goes down and then flattens out. When you've got something like even a bit of an antibody, an Fc region from an antibody, or an intact antibody, you've got no chance of increasing the flow rate to speed up your method without losing efficiency of your separation. So the best thing to do with your proteins is to try and optimize the flow rate, and slow tends to be better. So ideally, you don't want to increase the flow rate too much because you will get a much worse peak shape if you're going too fast. proteins like to go much more slowly in their diffusion. Here you can see some optimization of flow rate when looking at peptide mapping. So in this case, we're looking at peak capacity in separation, and that is a measure of how many peaks I can get into a separation window. So the best peak capacity is the highest one, and in this case, going at 0.35 mils a minute gave us the highest peak capacity in our separation. So it's worth considering your flow rate when you're running your method development. Also want to look at pH. For proteins, you've got that PI, so you can work at above their PI or below their PI, and you'll get different ionization on the column. You can see most of the applications are at low pH, but you do get significant changes in selectivity if you change the pH. Here we're looking at 2, 4.4 and 6.5, and that's just based on the fact the column can only go up to pH 8 or pH 7. So if you have a column that goes to a wider pH range, obviously you could look at a different pH. So you can see big differences in the peak spacing and the peak elution order based on changing the pH. We have here also consideration of the gradient, and I'm not going to dwell on this because I've already said flatter, less steep gradients are better for increasing the peak capacity in a protein or peptide separation. So do try and go for a flatter gradient slope when you're working with these types of molecules. If you have to speed up, like I know I do, then you can if you've got enough separation. If you've got enough resolution between your peptides or your proteins, you can try and speed things up. But that's the only time that I'd suggest increasing that gradient slope, because most of the time you're going to get a better resolution, a better efficiency if you've got a flatter gradient slope. In fact, for peptide mapping, flattening it here, you can see to 30 minutes gave us the highest peak capacity. So it does really help to improve your separation. And if you want to go even further, you can couple columns together. So longer columns give you a better separation too. But what you pay with is time. So here we've actually put 3 columns in sequence, and then that's improved our separation a lot. You can model this using our gradient translator tool, which you can download from the MatMod website as well. So that will help you to look at how you can translate those methods and make sure you're maintaining the same gradient even with columns coupled together, so longer columns. So finally, just a very brief mention of temperature. And temperature is something you should consider. It affects both the protein structure and also the method. So do consider how it's going to potentially denature your protein. Here, for example, we've got separation of a monoclonal antibody, and you can see a much better valley when we've gone up to 60 degrees compared to when we're running down at 30 degrees. It has denatured the protein, it's unfolded, but it's given us a better separation. And we do that a lot for things like the oligonucleotides as well. Whereas for peptides, just consider temperature and how it's going to affect your protein, the structure of your protein, or potentially the structure of your peptide if it's got any tertiary structure as well. So with that, thank you very much for listening. I have a couple of polling questions at the end, which I'm going to click onto. And then we should have maybe a couple of minutes of questions at the end. So on your screen, hopefully now you can see the first polling question. I'm just going to read that out and give you a bit of time to answer before we go into the live questions. Would you be interested in learning more about ACE bioanalytical columns? Yes or no? And most of the applications I've talked about today, we've run on the ACE columns. either the 100 angstrom or the 300 angstrom. So we've got a lot of experience, and obviously MacMod have as well with those. The second and final question I'd like you to consider, please, is do you feel like you learned something new about biomolecule separations or method development today? And I really hope you have. I've tried to cover probably a bit too much in the hour, but I think we've got some questions, and we can always contact you afterwards if we run out of time today. So with that, I'm going to pass you back to Catherine and see if we have any time for questions. Sorry.
Thank you, Gemma. That was a great presentation. I hope our audience will stick around for a few minutes. We're going to go for another 10 or 15 minutes to answer your questions. First question, if a shallow gradient is helpful, would isocratic multi-step method have a similar resolution?
Isocratic multi-step. Potentially, I think you do need some sort of increase in acetonitrile to get the proteins to come off of the column. If it's got multi-steps, then I'm guessing there will be some small gradient parts in there. It might be quite hard to optimize, I would say, if you're going to work with something like that. And I'm not sure we've done much work with isocratic, to be honest. We've really tried to stick with low percentage increase gradient methods.
Okay, thank you. Next question. You mentioned that TFA is commonly used for LCMS, but I read in the literature that TFA can form ion pair in gas phase, which will suppress the ionization. So I thought formic acid was more popular. Is that wrong?
No, it's not wrong. TFA is not great for mass spec. It does cause ion suppression. We can get loss in sensitivity, especially if it's in the area you're interested in. Formic acid will work better for mass spec, yes, but it does not give you the same improved peak shape. I know there's been a lot of work more recently with DFA, so difluoroacetic acid. So that could be another alternative. There's a lot being published in the literature sort of currently about DFA. So if you can't get to really low concentrations of TFA, then I'd consider DFA as an alternative. Sometimes formic acid will be okay. We just haven't had a lot of success actually with the proteins we've been working with formic.
Okay, great. Next question, what are some strategies to minimize the carryover of large proteins?
That's a very good question. And actually, I did have a slide on carryover in my original slide set, but it was obviously too long already, so I took that one out. Carryover is a right pain, to be honest, when it comes to proteins. You need to try and work out where it's sticking. Some columns it will stick to. Sometimes it will stick to the stainless steel. Sometimes it's very hard to work out where your protein is sticking on the system. So lots of the time you do have to change the column, I would say. Maybe looking at bioinert systems if you're having real issues with carryover. Adding extra blanks, lowering injection volumes. It's a challenge if you've got something that is really giving you loss in concentration because it's sticking to the column. Sometimes you can flush the system and remove them as well. But yeah, it's a challenge. Carryover is one of the biggest issues, I would say.
Great. Lots of great questions coming in here. What does the great audience. What does the TFA modifier do to the column? Block the free silica sites?
So yeah, it will definitely for lower purity columns. It will sort of cover any active sites that are still on the column. That's why it's quite good to have high concentrations with those sort of early generation low purity columns. It also will ion pair a bit with your sample, so it's acting as an ion pair in the system. It's quite hard to remove it from the column, not as hard as things like HFIP, but you will tend to get a build-up of TFA on the column as well. So it could potentially change the column slightly in the long term as well.
Okay. Next question. What do you mean by long equilibration times? Are you referring to priming the column or analyte equilibration with the column?
Okay, so in terms of long equilibration, reverse phase, we can put our column on the system and within sort of 5 minutes of running our mobile phase through, we've got a steady baseline and we've got a steady back pressure. When we look at ion pairing, it tends to take a much longer time for the ion pairing to settle. So you don't get a flat baseline and you don't get consistent chromatography. You tend to get a drifting baseline because it takes such a long time for the ion pair to sort of, I was going to say equilibrate again, get to a consistent level within the column that's reproducible. And it's the same with Hillock. So in Hillock, you have to flush the column with sort of 20 to 50 column volumes before it's going to be in a steady state that you can run your first injection. Whereas a reverse phase column, you can put 10 injection volumes down there and it'll be fine. So that's what I mean by equilibration.
Okay, terrific. Next question. How do we know which ion pair reagent is suitable for my compound?
Oh, that is $1 million question, and I think I would do very well if I knew the answer to that. Trial and error, I'm afraid, a lot of the time. If you've got a screening system, you tend to not want to put ion pairs on there because they do modify your column. You can look through the literature and see if anyone's run anything similar to your compound previously. There's things like the oligos, then HFIP, TEAA are very commonly used ion pairs, but there's not really a good guide out there that I've seen, if I'm honest, to select the right one.
Okay. A few more questions here, a couple more minutes. What hydrophilic groups are on the ACE columns used for oligonucleotide separation by ion pairing?
Okay, so the ACE oligo column is I think the functional group is actually proprietary. So it is an alcohol-based stationary phase. So it's going to be very similar to things like a C18, but the actual specific stationary phase is proprietary. So I can't share that one.
Okay. Next question. Is there a mode of chromatography that you recommend for the analysis of biomolecules? In other words, RPLC or Hillock?
It depends what you want to do and how hydrophobic your proteins are. If you've got more hydrophobic proteins, then reverse phase is going to be best for you. If you've got more polar, more hydrophilic proteins that are coming out early, when you try them on a C4 column, for example, then I'd move to Hillock because you'll get more retention and therefore more chance of separating them.
Okay. Next to last question here. What happens from a regulatory perspective as analytical techniques improve in sensitivity and ability to detect what were previously undetectable contaminants and variants of previously manufacturers and currently sold biologics?
That is a very good question also. As we improve the chromatography, often we see more things. Often, if you run a method that was developed on a column from 1970, now you run it on a new type of column, you'll find something else in that formulation. And if you're the person that finds that, then yeah, you've got a lot of work on your hands. So from a regulatory perspective, most of the methods we're going to be following are validated methods. They have got a specific column written there. It's only when you revalidate on perhaps a UHPLC system or you change the column to a newer generation column that you start to see these extra things in there. So if you find it, you have to characterize it, you have to go to the bodies and tell them But if obviously you've got maybe some examples of historic material, historic batches, you can often prove that they've been in there all along. If you've got an older batch of your drug, for example, and it's just now you found it with this different method. If it wasn't there in one of the older batches, then that causes a lot more regulatory problems, of course.
Okay, terrific. Thank you. I think that's all the time we have today. Thank you again, Gemma, for a terrific presentation, a lot of information in there. And thank you to our participants for being a great audience and sticking around for a few extra minutes for questions. Be sure to check CNEN or CNEN Online for information on the next edition of CNEN webinars. And thank you On24 for technology and production services. And thank you MacMod for the sponsorship that made this interactive webcast possible. For CNEN webinars, I'm Catherine Dold. Goodbye.